Okay. We will, I'm not sure what that was. We will now finish off thinking about multivariate stuff with thinking about multivariate analyses that are based upon a distance matrix as opposed to a correlation matrix. The good news is that if you understand how PCA works and RDA works, these distance-based methods are pretty much the exact same thing. There's only a few very small, subtle differences that we will talk about. And so we're going to jump in. We're going to think about multivariate analysis based upon distance matrices, which is a pretty common type of data to have, especially in ecology and evolution, as well as other fields that fall underneath that umbrella. So, if there's microbial people out there, maybe you use unifrac distance. Well, that's a distance matrix. It's a pairwise distance matrix, and it tells you something about the dissimilarity among your samples. So we're no longer going to be dealing with a square matrix of raw data that are converted into a correlation matrix. Rather, we're going to be dealing with data that are explicitly from the onset, some type of dissimilarity structure. So let's get started. Here we are in the wonderful world of distance-based ordination. So the distance-based analog to a PCA, principal components analysis, is what's called a principal coordinates analysis, or Picoa for short. Majority of the math that's happening under the hood is the same as with a PCA. We're going to calculate eigenvalues and scores for each site. The only piece of information that is lost when doing a principal coordinate analysis is you don't have a formal eigenvector. So you don't necessarily have the loadings. Now, if you search online and you do some reading, as it were, there are methods for determining what the likely loadings are by looking at the correlation of your raw data to the PC scores, essentially, to finding out, and that's a way of finding out which of your the components that went into making the distance matrix are necessarily the most important. But usually, there are better ways to extract that information that are not part of principal coordinates analysis. So let's start with principal coordinates analysis, and let's just think about it as a exploratory data analysis tool, just like we did for principal components analysis. So one thing that I found very satisfying when I started exploring principal coordinates analysis is that if I were to take my raw data matrix, the one that we would put into the PCA, if I were to Z-transform all the columns, right, which means that that's going to be the same thing as doing it on the correlation matrix if we were in a principal components analysis. But if I were to Z-transform all those columns and I were to take the Euclidean distance between all of the points for each sample, I would get the exact same answer that I got doing a principal components analysis. And so I want to start here just so that way you don't think that there's something magically different about principal coordinates analysis as opposed to principal components analysis. Again, the important difference is that principal coordinate analysis is based upon the input being a distance matrix, whereas a principal components analysis, the input is the data, which then are converted either into a covariance or a correlation matrix. So let's start with the principal coordinates analysis. And we'll go back to the butterfly wings. And I will go ahead and I'm going to use this function called vegdist. Again, vegan is a package that was built originally for people that do plant communities. So there tends to be a lot of plant words in it. but it's just simply a function that's going to give us a distance matrix. And I'm going to ask for it to kindly give me the Euclidean distance. Okay, so again, it's just multivariate space with all of those different measurements. And I have individual 1 here and I have individual 2 here. And it's just the Euclidean distance. It's how, as the bird flies, right? We're borrowing from Euclid. It's just simple linear distance between two things. And so let's go ahead and do that. And ignore my little graphical warnings. See? And then I can look at the very top of this distance matrix. And so the distance in that multivariate space of all of those measurements between 1 and 2 was 1.4402. The difference between 1 and 3 was 0.739886. Okay, so all this is a diagonal matrix where we're thinking about the Euclidean distance between points in multivariate space. This will give us the exact same results as doing a principal components analysis on the raw data. So I'm going to go ahead and I'm going to run it as an RDA. And then the function that we'll use for doing Picoa, and there are packages that have functions called Picoa, but I'm going to just stick with using vegan because I find it intuitive and easy, is I'm going to use a function called dbRDA. dbRDA stands for distance-based redundancy analysis. And here I'm going to say that I only want the intercept, which means that it's going to give me a principal coordinates analysis. So now I've analyzed the exact same data. One of the data here, or the data here for this object, BWRDA, that is a principal components analysis on the covariance matrix. And then here, BWPCoA is a principal coordinates analysis based upon Euclidean distances among all of those points. And so I can go ahead and I'll put that one into the working environment. And then we can have a peek here at what's happening. So with that object, here's our eigenvalues for the unconstrained axes. This is principal components, right? This should all look very familiar. This is principal components on the covariance matrix. We know what the inertia is. It's just the sum of all of the variances together, right? All of these numbers, well, 1 through 24, if we were to add them all up, they would equal 1.09 or 1.909. And so that's for doing it as a PCA. And now we can do the exact same thing doing it as a Picoa. And the numbers are identical. The eigenvalues are identical. The inertia is identical. And it's because it's doing the exact same thing. The exact same thing. The only difference is here they're calling them MDSs, which stands for multidimensional scaling, whereas here they're PC, which stands for principal components. But you can see that they're just branded as having different names, but they are the exact same thing. So if you were to plot the PCA that we got from this line here, if we were to plot that, and then we were to plot this, the plots would be absolutely identical. And so then, as I mentioned, if we were to Z-transform the data, the columns, and then take the Euclidean distance of the Z-transform data in a Picoa framework, that will give us the exact same answer as an RDA on the correlation matrix, right? Or a PCA on the correlation matrix. So here we are making a principal components analysis. We're saying scale equals true. So that is going to be on the correlation matrix. Everything is Z-transformed, right? Because all correlation is standardized covariance. So I'm going to go ahead and I'm going to run that. And then I'm going to create a new butterfly wing object, because I don't remember if I had previously, where it's going to be Z-transformed. And so I'm going to scale the butterfly wings, and I'm going to do that by just saying scale butterfly wings. That's going to automatically scale all the columns, so everything's going to be Z transformed. And then I'm going to create a Euclidean distance matrix using that VegDist function. And so this will give me a pairwise distance matrix that we can look at just to make sure that Jim's not telling tall tales. There it is, right there. Okay, so now we're in Z-transformed multivariate space, and this is the Euclidean distance amongst, this is 1 through 2, this is 1 versus 3, 1 versus 4, 3 versus 2, and so on. It's a diagonal matrix of pairwise distances, Euclidean distances based upon Z-transformed data. And if we were to put that into the dBRDA function, which again, by saying all we want is the intercept, that is identical to saying we want a Picoa. So we can do that. And I think I ran that, but okay, so now let's take a look at that object. That's that right there that we're looking at right there. There is the principal components analysis on the correlation matrix. And what we are looking at right here, that is the principal coordinates analysis based upon the Euclidean distance of the standardized data. And again, we get the exact same eigenvalues. And so if we were to plot those out, we would get the exact same plot. They would look no different. So that should be somewhat satisfying, right? Because like once you know that, it's like there's no difference between the two. Why in the heck are we wasting our time? Which, I think somebody could argue is a really, really good point. But hopefully it's satisfying that they're both doing the same thing. So why would anybody want to do a principal coordinates analysis? The main important reason is that oftentimes we're dealing with data that it is not appropriate to take the Euclidean distance because the Euclidean distance doesn't really mean anything. And so in particular, I'm imagining, let's imagine a large matrix of like microbes, the number of, 100 different species of microbes across 100 different soil samples. In general, you wouldn't want to take the Euclidean distance between those because it's unclear what it means to Z-transform all of those columns. And then it's even more unclear how you interpret the Euclidean distance based upon counts of the number of things in a matrix. And so in ecology, for example, people use things such as Bray-Curtis, which is basically a way to describe how dissimilar two samples are based upon the number of shared species that they have with one another and the number of unique species each of those samples have. And Bray-Curtis is often the standard one that people use. I'm going to revisit that in just a second because it might not be the best one, but it certainly is the one that is the most commonly one used in the literature. Similarly, if you do like population genetics, you could have pairwise FSTs as being some measure of population dissimilarity. And so that is the really nice advantage of doing a Picoa, is that we can be really flexible about how we want to think about the dissimilarity. And there's a whole chapter in this book about all the different measures of dissimilarity. And even, let's see, I'm going to go off script because I have a script. Let's see here. I have a script. Question mark. VegDist. Remember, we used that function to say, hey, we want Euclidean. But it actually has, as a default, it does Bray, which is Bray-Curtis. But you can see, if you look down here, there's all these different distances. Euclidean, Manhattan. Manhattan distance is sometimes really useful when dealing with like chemistry data. If you have very complex chemistry data, a lot of times people like to use the Manhattan distance. But there's all of these different types of distances. And really, it's kind of the onus is on you as to figuring out what dissimilarity metric is the best for your particular question. And again, there's a really nice chapter on that in this book that's available to all of us on the library. So that's going to be a nice advantage of being able to do principal coordinates analysis. So as a start, just to kind of think about the behavior of these, I'm going to create a fake data matrix, and we can look at it, where I have 6 different sites, and I have 7 different species. And you can see that there's differential abundances of all of those species at the different sites. I'm going to ignore issues right now, such as whether or not you should be doing rarefaction. I think those are really important conversations to have, or certainly to think about, but it's well beyond the scope of what we're trying to accomplish here. So we're going to just assume that the data that we have are totally legal to analyze how we want to analyze them, and this is what the raw data look like. And so if I wanted to ordinate those points, the first thing I would want to do is come up with a dissimilarity matrix. And so in this case, I'm going to choose the default, which is Bray-Curtis. And I'm doing that for no reason in particular, except it's a common one. And it really is a common one. I would argue that at least 80% of ecology papers use Bray-Curtis dissimilarity over any of the others. And so I'm going to go ahead and I'll run that veg.dist. And now we have a pairwise dissimilarity matrix. So this is the dissimilarity between site 1 and 2 versus the dissimilarity between site 1 and 3. The lower the number, the more similar those two sites are. The higher the number, the more dissimilar those sites are. So sites 1 and 5 are way different from one another compared to sites 1 and 3, for example. And so now we can take that dissimilarity object and we can put it into the dbrda function. I probably should mention this as well, just make a little mental note. dbrda is a relatively newer function in vegan. In the past, the function that people would use to do the exact same thing was called CAPScale, which stands for Canonical Analysis of Principal Coordinates. They're almost identical to one another. They differ in pretty esoteric ways as to how they deal with the eigenvectors that are extracting. In my personal experience, I've never gotten dissimilar answers using either of those functions, but now people have gone to using dbrda. In fact, if you hit $dbrda, you jump onto the CAPScale function, okay? And so they're really similar to one another. They take the exact same arguments. They just differ in very subtle mathematical ways, which are described here. But we don't need to worry about getting into the weeds as to how they're different from one another. I mainly mention that because there's a lot of super helpful tutorials online. And if they're 4 years old, they might be using CAPScale instead of dbrda. I guess at this point, they would have to be about 6 years old. But hopefully you get the spirit of my point of digressing, as it were. So let's go ahead and we'll do the principal coordinates analysis on our Bray dissimilarity matrix. So we'll say dbrda, our distance matrix, (1), which means just do the intercept, which means don't do anything, which means just give me the principal coordinate analysis. So I'll go ahead and I'll do that, and I'll ask for summary. I have a note here to ignore this warning message. The reason why you're getting that, or I'm getting that now, that warning message is that we did an intercept-only model, and it's just warning us that it doesn't have anything to identify the LHS, which is the left-hand side of the column. It doesn't matter for our purposes at all. Remember that warnings are not errors. They're just letting you know that R is assuming some things, and they're letting you know what it's assuming. And so we have our we have our principal coordinates analysis. And we could go ahead and we could plot that. Let's just plot it and take a look at it. Okay, and so you can see sites 1, 2, and 3 are all clustered here, and then sites 4, 5, and 6 are kind of all spread out. They're clearly different from one another. So that's like a first step of feeling kind of good at it. If we wanted to do best practices, we could do something like that, or maybe even replace these with the actual, like with dots if we wanted to. But that is the principal, that is basically the principal coordinates analysis. And we're capturing a ton of the variation. But it's not terribly surprising that we're capturing a ton of the variation because we only had 6 sites in this example, right? And so the first 2 axes are explaining 90% of the variation. In real life, you're never going to have only 6 samples with 8 species or whatever we had there, 7 species. And so it's never going to be quite that high. But we interpret the rest of it the same way that we would interpret the output from a principal components analysis, except here it's based upon the a dissimilarity matrix, and we happen to have used the dissimilarity matrix here where it's Bray-Curtis. So to give you an idea of maybe how this would look in a more realistic framework, it's not fully realistic because I'm simulating some pretend data. So I'm going to simulate some data here. And this is going to resemble more of what like a real community matrix would look like. And so I can look at the dimensions of this. There's 400 rows, so I had 400 different samples, and there's 489 columns. So there's 489 different microbes. And we could look and see what that data that I cooked up looked like. Let's just look at a small subset of it. First 10 rows, first 10 columns. You can see it's pretty sparse. There's lots of zeros, lots and lots of zeros. And actually, if we look at the head of dat.micro, you know, here's some pretty common species that are found everywhere, but there's a lot of zeros. And especially with these rare species, there's lots of zeros. And, you know, right now we can think of why a principal components analysis would probably not be appropriate with this data, because it's based on the correlation, right? And when you're doing a correlation, you really want the mean to mean what you think the mean means. And if you have that many zeros and you have no negative values and you have this long tail of positive values and it's count data, it's probably not appropriate to think about it as a correlation. And so the data that I've then, so again, the data that I've cooked up, it resembles the kind of data, and I'm only picking microbial data just mainly because I've been dealing with a lot of microbial data lately. But we got 400 different samples. We got 489 different species there. And you could think about these as reads, the number of reads. So we're going to assume that that's some measure, quantitative measure. And then we have 2 different populations that maybe we're interested in knowing whether or not these 2 different populations are different from one another. And by population, I mean, if these are microbiomes, maybe it's from the, you know, the gut of 2 coexisting fish species, or maybe it's, you know, some kind of a microbiome between diseased and non-diseased. You can make up any kind of story that you want. You can imagine why people might be interested in understanding how the microbiome differs between 2 things. And so I've just created this long vector of identifying whether or not it's in population 1 or population 2. So the first step I'm going to do is I'm going to create a dissimilarity matrix. And here I'm going to just change it up a little bit just to plant some seeds in your head if you ever are dealing with these types of data. For VegDist, instead of using Bray-Curtis, I'm going to use Jacquard. And then the first really important thing to realize, and this you're going to love this is really nice and frustrating, is that what I've typed in right there isn't going to actually give me Jacquard. How's that for convenient? I asked it to give me Jacquard, and it's not going to give me Jacquard. That is because formally, not formerly, but formally, Jacquard is based upon incidence data. So presence or absence, that's the only thing that Jacquard takes. So we would have to go into that matrix and convert all non-zeros to ones. So it would just be zeros and ones. It's either present or it's absent. There is a continuous analog of the Jacquard matrix that should actually probably be called Ruzicka index. And if you give the VegDist function data that is not zeros and ones, it's going to assume that those numbers mean something more than presence absence. And so when you ask VegDis to give you the Jacquard dissimilarity, it's actually giving you the Rosica dissimilarity. If I wanted it to do the true Jacquard, the argument I would use is binary equals true. And then that will tell the function to convert everything to zeros and ones, and then it will give me the true Jacquard index. But I'm not going to tell it that. I just want it to give me the continuous analog of Jacquard. I'm not really sure why I kept saying continuous, but I did. What I meant to say was quantitative. So traditional Jacquard is an incidence-based, and then you could think of the Rosica as being a quantitative analog, not a continuous analog. But I made a mistake, but now it's fixed. Back to our show. So why am I choosing to do that besides confusing you with all these dissimilarity metrics that we haven't talked about? The reason why I'm doing that is that I like the Rosica index personally better, as well as other people do, including Oksanen, the guy that wrote the vegan package, because the Rosica index is what is called metric, meaning that it will satisfy what is called the triangle inequality, where the lengths of any two sides of a triangle have to be greater than the third side. So you could imagine if you have 3 points in multivariate space using a dissimilarity metric, you expect that it'll satisfy the triangle inequality. That is not always the case with Bray-Curtis. And so in one of the help files in vegan, Oksanen even says that although everyone likes to use Bray because that's what people have used for 40 years, 50 years, 60 even maybe, that it's probably more appropriate to use the continuous Jacquard because it is metric. Esoteric point, again, it's not wrong if you use the Bray-Curtis. That's what 80% of people probably use is Bray-Curtis. I just wanted to kind of point that out. So okay, so I'm going to do my Rosica dissimilarity, which I'll call Jacquard because that's what they're calling it, so I'm going to call it that. And so I have my dissimilarity matrix, which we can see the very top of it right here, 1 versus 2, 1 versus 3, so on. And then I'm going to color the populations red and blue so I can tell the differences between those two when I plot them. And I can start with, a little aside. This little thing I typed here, I'm going to type it right now so it's in your notes because I'm about to post these. I'm going to say this just increases the print buffer in the console. Basically, I don't know if you've noticed yet because we probably haven't dealt with any data sets that are big enough, but there's a limited amount of information that this window will hold on to, and you can't go any further in the history than that. I'm stuck there. And so by saying max print to a higher number, it's just going to allow me to print out more things in the console window so I can find my way around, because these are going to end up being really big objects because, again, we're dealing with 400 rows, so there's a lot of information that's going to be there. So let's do it as a Picoa, and then we can look at the summary of the Picoa. And what do we want to pull out from here? First, you can see why I did print max, because I'm all the way up here. And so what do we have? First we have, and again, this is a Picoa. This is not a constrained axis, so there is no RDA axis. They're all just multidimensional scaling axes, which we can interpret the exact same way as PC axes, essentially. And you can see the first one's explaining 38%, the next one's 12%, the next one's 5%, and so on. So the first thing I want to point out here is that the cumulative proportion has these NAs in it. And those NAs are there because a peculiar thing, if you will, happens when we're not dealing with Euclidean distances, where eventually some of these axes, I'm going to get there. It doesn't happen till way long time, but there it starts. So on the 227th axis, now we have something that says IMDS. And it says I MDS because it's an imaginary number. And it's an imaginary number because our eigenvalue is actually a negative value. Remember that in principle, our eigenvalues are supposed to be variances. You cannot have a negative variance, which means that this must be an imaginary axis. And so because we have that imaginary axis, we're unable to calculate the cumulative proportion of how much variation each axis is describing or explaining. So this has really never been a problem, at least in my experience, because by the time we get to the imaginary axis, we are explaining such an infinitesimal little portion of the variance that who cares, right? We're usually only interested in the first few handful of axes that are explaining most of the variation, not things that are explaining, 1,000th of 1% of the variation. That is not terribly interesting. And so, but if we wanted to fix that problem, which maybe we do, we could add an argument to the dBRDA function called add equals true. And this fixes the problem of the negative eigenvalues simply by adding a constant to everything, so that way we have no negative eigenvalues. And so if I do that, now you'll notice that I have the cumulative column there, okay? So 18% for the first axis, 5.8% for the next axis. That is the accurate representation of the amount of variance explained, as opposed to doing it the proportion explained when you have imaginary axes. Because then what you do is you sum up the total variance and then you start subtracting from it, and it will tend to inflate the proportion explained. So in my impression is that it's a good idea to say add equals true, especially if you want to start interpreting the amount of variation explained by your axes. That said, add equals true. If we were to save the PC scores from where we didn't say add equals true and look at the PC scores where we did say add equals true, the correlation between those two is 1. So we are arriving at the identical solution, and actually we will arrive at the identical appearance of our plots. There it is where we have the imaginary axes that we were maybe worried about, and here it is when we added that constant by using that add equals true function, and you'll see nothing has really changed. The only thing that's changed is if you look at the axes, they've shifted over a little bit. But it doesn't matter because we don't really directly interpret, -1 or -0.5. We don't really directly interpret those anyways when we're looking at the ordination. And so the plots themselves are identical. The only thing that has changed is the values on the axis, and that's because we asked the dBRDA function to add a constant to everything so we wouldn't get those imaginary values. So but once we have that, that's really the only differences that we need to think about with a distance-based redundancy analysis. And so if we wanted to know whether or not the populations were different from one another, we could do a dBRDA, where now our predictor are our populations, which again, it's population 1 and population 2. It's a real simple model, pop 1 and pop 2. So we go ahead and we run that, and now we could plot that if we wanted to, because why not? And you can see that the ordination looks a bit different, right? Now we only have one axis that is really primed to try to pull apart the two populations. This is just a multidimensional scaling. That is basically on the residuals. That's doing a principal component analysis after we've removed the variation due to our constraint. And here our constraint is population. And so you can see that it's tried to pull them apart on this axis. Ignore the fact that they're being pulled apart like this. Just think about the fact that the center of mass for the red looks to be somewhere over here, and the center of mass for the blue looks to be somewhere over here. So it did the best job it could at pulling apart population 1 and population 2. And then as before, we could go ahead and we could do the hypothesis testing on it. So we're going to do the same kind of a permutational ANOVA that we did when we did it with just a straight up RDA. The only difference here is that we're doing it on a distance-based RDA. For our purposes of making this not go forever, I'm going to just restrict the number of permutations to 999, which is the default. And I'm only doing that because now we're into a land where we are dealing with a pretty large matrix, and it takes a couple of seconds for the computer to think, right? So now it's working really hard, and it's doing 999. Didn't take that long. But I mean, just to give you an idea, if I did 9,999, I can step up, fill up a cup of coffee, and it's empty. And we're still waiting patiently. And so you can imagine if I did it at 99,999, it would take a long time. And actually, when you have real data, where you maybe have like thousands of rows, thousands of samples, this type of an analysis, if you were to do it 99,999 times, it might take as long as 12 hours to complete the analysis. Which is why it's always a good idea to set your seed so that way you don't have to do it a second time. You already know the answer the first time you do it. But, okay, so we've done it here. We've got 9,999 permutations, and there's the p-value associated with that. So we would say that there is a difference between the two populations in their microbiome. And again, just as with the previous examples that we've been playing with, there could be more information in there, right? I could have something else. I don't have it because I simulated it, but you could imagine you could have a moisture gradient that went along with that, or a soil pH gradient, or whatever it might be that went along with that you could also put in the model. And if you have more than one term in the model, that's where you would have to say phi equals margin. And then you could run it by the margin, and you're going to get the exact same answer, because we only have one effect in the model. It is always the last thing in the model. So yeah, that's how you do a DBRDA. I mean, again, if you understand how RDA works, DBRDA is just reminding yourself that you're dealing with a dissimilarity structure, a distance matrix, as opposed to dealing with a matrix of raw data that then you either look at the covariance or the correlation among those two. I guess one last thing that I'll mention with permutational type ANOVAs, and this is more of trivia than it is anything. You've probably noticed dealing with other programs in the past that they always tend to be something like 999 or 9,999 or 99,999. The reasoning behind that is really just to make the math easier if you were to do this by hand. I am pretty convinced that it really goes back to when people would have to calculate these p-values by hand. Because the idea is that we've done 999 permutations. Our 1,000th observation is our actual observation of our data. And so we're asking where that 1,000th observation falls outside of all 1,000 of them, which is the 999 plus our actual observed. But I don't know if anyone's ever noticed that before, but generally when permutations happen, it's always 9 is always the last answer or last number. But that's, I think, just for the ease of being able to calculate p-values without having to do more complex division by hand. And so doing 1004 is totally fine. You could pick any number you want. General convention, standard convention at least, is to have it something 99999. Okay, let's take a quick break, and then we'll talk about one last thing with distance-based ordinations. The last one that I wanted to mention is one that is different from all of the other ordination methods that we've talked about thus far, and it is what's called NMDS, which stands for non-metric multidimensional scaling. And NMDS differs from the other ordination methods that we've looked at, in that the other ordination methods that we looked at could be categorized as analytical, meaning that there is a solution. There is only one solution. Regardless of who on the planet is looking at your matrix, they are going to get the exact same PCA as anybody else, assuming that they do the arithmetic correctly. NMDS is what's referred to as numerical. And basically that is a way of saying that the computer is going to try to figure out a solution, but it's not going to always come up with the same solution. And so NMDS is used frequently with dissimilarity objects, kind of like the microbial data, the fake microbial data that we were playing with, is be a common place where you often see NMDS plots. And how NMDS is working is essentially you are going to tell the computer how many dimensions you want to ordinate into. And it's easiest to think about it as 2 dimensions, because we can think about what a plot looks like. And so what happens behind the scenes in the computer? So you have this distance matrix. And you could imagine that you could ordinate this distance matrix based upon their dissimilarities and however many dimensional space it is. And then what we're going to try to do is take all that n-dimensional space, however many dimensions we have, and project them, in our example here, to a 2-dimensional space, because we can ask the computer, give me 2 axes. And what it's going to do is it's going to rotate that data in such a way that when it projects it on a 2-dimensional surface, that it is retaining the rank ordering of differences among all of the points across all of the samples. So if we were in a classroom, which we're not, and I don't have a spotlight, the metaphor that, well, actually, I guess it's more of a simile, because I'm going to say like. The simile that I like to use for it is like, imagine if you had a bunch of 3-dimensional points, right? So we're only going to deal with three dimensions here. And it looks kind of like a ball, right? Like a big cloud of points. And then you knew the position of each of those points on this ball of points, and you knew what the differences were among all the points. You knew the rank ordering difference. So you knew that these 2 points were maybe the closest to one another, and then these 2 points were the second closest to one another. And you kind of knew how everything was dissimilar to one another in a ranked setup. And you have this big cloud of points. Now imagine putting a spotlight through that cloud of points, and you're projecting it then on a screen like this, right? And you're rotating it around. And what the computer is going to try to do is keep rotating and tweaking that around until it does the best job it possibly can do of taking this cloud of points and projecting it on a 2-dimensional surface, so that all of the points that it's projecting are as close as possible, or at least as close as the computer can come up with in that iteration, of putting all of the points is clustered as nicely together as possible while doing the best possible job at maintaining the rank order differences between those. And so it's a projection, which means that it's not how we think about the kind of eigen-based methods that we've been talking about, where you can actually extract information out of there and think about what is the most or least important, and what percentage of the variation is explained by a given axis. None of that really plays into non-metric multidimensional scaling. It's simply a tool to project your multivariate data into the fewest number of dimensions as possible, while retaining the rank ordering. And so NMDS has a long history of being really popular in ecology and evolution, I guess. People used to do it a lot with genetic type data. because it was really the best game in town when it came to trying to ordinate a dissimilarity or a distance matrix. Now the methodologies associated with principal coordinate analysis are there, and I would argue that you get more information out of a principal coordinate analysis because, first of all, there's only one solution, and second of all, you can actually interpret the eigenvectors. You don't really have anything analogous to that with a non-metric multidimensional scaling. So, historically, one of the weaknesses or constraints with non-metric multidimensional scaling is that it was computationally intensive. It might take hours upon hours to come up with an ordination because the computer is literally rotating around this n-dimensional thing and trying to find a way to project it on a two-dimensional surface where you're maintaining the rank differences. Computers are faster now, so that's really not quite as much of an issue. It can still, with really large data sets that are ugly, generally the uglier the data is, meaning that the more difficult it is for the computer to come up with a good solution because there might not be a good solution. Those can kind of drag on and be slow, but for the most part, computers are fast now, so that doesn't matter. The other constraint or concern or something to be aware of with non-metric multidimensional scaling is that at the end of the day, you really have lost the information about the magnitude of the differences. Because the first step of an NMDS is its rank transforming everything. And so that is also an important thing to keep in mind when considering doing an NMDS. Now, me personally, I'm not a big fan of NMDS. I will come out and I will say that. I even have this t-shirt that says every time I see an NMDS plot, a small part of me dies, which is probably a little hyperbolic, maybe a bit hyperbolic. And it's not so much NMDS itself. NMDS, I think, is incredibly useful as a way to kind of just look for generalized patterns in your data. I think that the reason behind that snarky shirt is I don't like how they're used because they're often used in conjunction with things that have nothing to do with the NMDS. So if you were to look in the literature, for example, in the microbial, especially the microbial literature, as well as just any community ecology literature, the common workflow is we did an NMDS, and then we followed up the NMDS with a per MANOVA. Now recall, per MANOVA is a loaded term because it means a lot of different things, but here I'm talking about the brand per MANOVA. I'm talking about Marty Anderson's per MANOVA, where it is looking at the distance of your data from the multivariate centroid to the overall distance to if you weren't fixed at those centroids, basically. So it's very similar to an RDA. My issue is, and I don't have any problem with per MANOVA. I don't use it very much. I use RDA usually because I find it more interpretable, but per MANOVA is fine. The problem I have with that workflow that like 80 to 90% of all papers do this. We did an NMDS and we followed it up with a per MANOVA, is that the per MANOVA is actually based upon a Picoa, a principal coordinates analysis, ordination. It has nothing at all in the entire world to do with the NMDS. The analogy I could think of is imagine if you were to do an ANOVA, but when you had a figure in your paper, you did the analysis and it was an ANOVA, but you show a bar plot based upon the ranks of the data. That would really not make a lot of sense, right? I mean, you did the analysis assuming that you believed what the mean was and the standard error was. Why don't you show me that instead of showing me the rank difference, but then do an ANOVA on the raw data? To me, there's just a disconnect there. And I don't know, maybe I'm just being a pedantic fool. I'm a fool. But it kind of drives me nuts because they're not showing the same thing. And as a consequence of them, that NMDS is rank-based, it's not necessarily true that points that are closer together in the plot are necessarily more similar to one another if you were to look at the raw distance matrix. And that's because the computer's doing the best job it can possibly do to impose a two-dimensional structure again, assuming that we want to tell NMDS that we want 2 dimensions, it's doing the best job possible, but it's not doing a perfect job. Okay. I mean, I suppose that you could cook up data where it would do a perfect job, but it's not going to do a perfect job. So you can't really trust the distances between those points because they don't necessarily mean anything. They might mean something, but they don't necessarily mean anything. And so that's why I don't necessarily like NMDS. I think mainly just because I think it's used incorrectly. But a lot of times it can be a useful way of looking at your data, especially if you have super dimensional data and you want to just look at an easy plot. Instead of looking at 5 PC plots with all the different axes, you could just say, I just want an NMDS plot with two dimensions and be done with it. So there is a way with NMDS that we are going to be able to ask, how good is our ordination? So how good of a job is our ordination doing at retaining the rank ordering of all of those differences in our input matrix? And that is what is, and there's a number of ways to do it. The most common way to do it is to use what's called Crustal Stress. And you might have seen this when you've seen NMDS plots or when you've done your own NMDS plots. Stress. And stress is a measure of how good of a job the computer did in ordinating at maintaining or retaining the rank ordering of all those points compared to your raw data matrix. And the general rule of thumb is that if stress is 0, that means that it's a perfect ordination. All of the distances between all of the points, the rank ordering is perfectly retained. Again, the Euclidean distance between those points still cannot be overly interpreted because the ordination itself is based upon rank transform data. But a stress of 0 means you're doing awesome. When you get up to 0.05, you're doing good. If stress gets as high as 0.1, fair. If you're between 0.1 and 0.2, poor. And if it's greater than 0.2, just fire a shotgun at a barn and that's your ordination because it's as good as random. It doesn't matter. And so that's how we interpret stress. One other thing to point out is that because it is an iterative process within the computer, depending on the program that you use, you might and usually won't always get the same ordination. So every time you run it, it's going to start with a different kind of random placement of points and then try to jitter them into the solution where it's done the best job possible at retaining the rank ordering difference. So also be aware of that, is that it's not always perfectly replicatable. Okay, so, but regardless, again, I don't have like serious hate for NMDS. I just think that it's important to be aware of what it's doing. So let's play really quickly with an NMDS just so we can kind of get familiar with how to interpret it and what it might look like. So I'm going to go back and I'm going to play with that that fake bacterial data that we cranked together when we were playing around with the RDAs. And so the function I'm going to use is in the mass package. It's called ISO-MDS. One nice thing about ISO-MDS is that it does have strict starting criterion as to when it starts doing the rotation and the ordination, so that ISO-MDS, in my experience, does always arrive at the same answer. But I have used other programs, like there's a program called Primer, which is popular in some circles. And at least the older version of Primer, every time you ran the analysis, you would get a different ordination. And the other important thing to remember with NMDS is that the first axis is not necessarily explaining most of the variation, and the second axis, the second most amount of the variation. The axes are totally arbitrary. There are ways that you can get in there and try to make it so the first one explains more than the second, but for the most part, the axes don't matter when I think about percent variation explained. So we're going to use this here, this ISO-MDS, and we're going to use that that Rusika dissimilarity index that we were playing with the RDA. And we're going to go ahead and we're going to run this. And you can see the computer's thinking and it's working and it's converged. So it's settled on that solution. So the first thing that we can look at when we're there is that our stress value, that's what this value is here, is we're in the borderline of okay, fair, but not great, right? We're certainly not at really good. Really good would be 0.05. Oh, I should mention this. A lot of programs, this one included, will multiply it by 100. So 0.05 is actually a 5. Should probably just change that, but I will before I hand it out to you guys. And so that happens, you know, a lot depending on what computer program you're using. They'll be different by orders of magnitude. And so here we have a stress of 11.17, which tells me that I'm not entirely thrilled by this ordination, but it might be interesting. So we can go ahead and we can look at it. So let's go ahead and we will look at it. Here's our ordination. You know, we can compare it against, there's our constrained ordination, there's our Picoa, when we did it just as a straight up Picoa, and then here's our NMDS. So they don't look that dissimilar, which isn't terribly surprising. They're attempting to accomplish the same thing. But again, it's a visual representation as to how these things might be different from one another. And again, it's really important to remember that just because these dots are super close to one another and these two dots here are slightly further apart, we really don't have any way of knowing the magnitude of how different they actually are, because this is all based upon the ranks. And so somewhere here, between here and here, is the furthest distance, I'm assuming. And so that would be the largest rank difference. And then some of these points here that are on top of each other are the smallest ranked differences. And you have all of these different distances, and that's what the computer is trying to maintain when it's coming up with this clustering. We can look at our object, and what do we get out of our object? So here's our stress. And then all of these are simply the XY coordinates for our plot. These are our points. That's all there is in the NMDS object. There's no how much variation is being explained. There's no loadings. There's no nothing. There's just the points and the stress. Okay. And so as I said, our stress is 11. We'd like it to be below 10. We'd love it to be less than 5, but 11 maybe isn't that good. So we can, by default, the ISO MDS function will say, I'm going to do it on 2 axes. But we could say, all right, well, let's do it on 3 axes. So now we're going to take something that's very multidimensional and we're going to project it into a 3-dimensional space. So we're going to have 3 different axes that we can think about. So we go ahead and we run that. And now we're in a pretty good territory, right? That's not horrible stress. And then we could go ahead and we could plot that. But now we'd probably want to have 2 plots. You know, the first axis versus the second versus the third, and maybe even the first versus the third, because there's no logical arrangement. For all we know, the third axis is the most interesting axis here. And we would know that. Whereas in a Picoa, we would know which one was. And so the stress value goes down the more we increase K. K are the number of axes. So now we're going to project it into 4-dimensional space, which we can't picture really what that might look like. But there is such a thing as 4-dimensional space. And now we have 4 axes that we could look at and plot. Okay. And that's all fine. So again, it's a fine way to look at the data. Just be really aware of exactly what it's doing. And it's not doing what principal coordinates analysis is doing. It is doing what NMDS is doing, which is based upon maintaining the rank order of differences. It has nothing to do with the actual values, because the first step of the process is to rank transform the data. And so to just get on my initial rant, oftentimes people would show their NMDS plot. They would show this in the paper or in a talk. Look at my plot, and then they would follow it up by doing a per manova. The function for doing per manova, which we didn't talk about, but it's very similar to RDA, very, very similar to RDA, what it's trying to accomplish. The function for that is Adonis. So I would just say my distance matrix is some function of my pops. So I could go ahead and I could run my permanova, and it takes a minute because it's doing the permutations. It's all there, and that's all I get out of that, but then I get a p-value. And so I would argue that it's wrong to show that figure and say that the blue and the red are different at 0.001, because this p-value here, that is all based upon a principal coordinate ordination, has nothing at all. It's not, it doesn't even know that this exists. But that is the normal workflow that a lot of times people do. And, you know, I'm over it. It probably, you know, I don't think anyone's making any huge mistakes by doing so. I'm not convinced that there's as much information here in the hypothesis testing framework than there is when you're just doing it as an RDA, or at least show me the Picoa and do the Adonis Permanova function on that. So that's Jim's very brief rant about NMDS. I don't dislike it as much as many of the rumors might imply, but be very aware of exactly what it's doing.